Welcome everybody to Duquesne University's annual Darwin Day. I'm very pleased that you chose this rainy Pittsburgh night to join us if you're in Pittsburgh or somewhere else around here. This is when I went and did the math, this is actually the 20th annual Darwin Day that we have held. So this is a kind of a historic occasion. Before I introduce tonight's speaker and the topic, I would like to go over just a couple items. The first and probably most important one is that there will be time for questions after this talk. If you want to ask a question, please enter the question in the chat box that you can see, I hope. And what we will do is when Dr. Rasgun finishes, my colleague, Dr. Michael Jensen-Seaman, will ask those questions for you, but we can take as much time as we need to get all those questions asked. So we are here to celebrate the life and work of Charles Robert Darwin, who is the founder of the science of evolutionary biology. Darwin was a model scientist. He took on many topics during his lifetime. He was an avid insect collector as a young man, mostly beetles, but many other things as well, which is sort of fitting considering that the topic of tonight's talk surrounds insects. If you're interested in Darwin beyond Darwin Day, I would strongly recommend visiting thepartnershipineducation.com, which is an NIH-funded program at Duquesne University. Several years ago, that partnership produced the Darwin Synthetic Interview, which is an app. You can ask Darwin all kinds of questions. He'll answer mostly in his own words, and it's free. So you can download that at one of these places. As I mentioned before, this is the 20th annual Duquesne University Darwin Day. Over the years, we have had many outstanding speakers covering a variety of topics like developmental biology, experimental evolution, domestication, genomics, human evolution. We had a COVID talk last year, and unfortunately, we're still in the middle of that. And this year, we're gonna hear about other emerging diseases that are insect-borne, not aerosol-borne. Before we go too much further, I also want to thank many of the people that were important in pulling this event off. Primarily, I'd like to recognize the sponsorship of our dean, Dean Ellen Gawalt. The deans of the Bear School over the years have been instrumental in supporting this program. Also, the people that work in the dean's office, Amy Walters and Jean Stone. I'd like to thank my colleague in the Department of Biological Sciences, Dr. Michael Seaman, who does, he and I both organize this and do all of the behind the scenes work. And of course, our graduate students that do much of the work as well. And before I introduce tonight's speaker, I would just like to point out that there is a pretty solid link, maybe unrecognized link, between insect transmitted diseases and the history of evolutionary biology. And it goes all the way back to the founders. Darwin himself, as a young man, spent a lot of time on the Beagle voyage in and around South America. When he got back, he was struck with a chronic disease that affected him his entire life. It kept him at home a lot, probably kept him writing, maybe more than he would have done. People have speculated that perhaps he had Chagas disease, which is an insect-transmitted disease. It's a protozoan disease that's native to South America. The other person responsible for discovering evolution by natural selection, Alfred Russel Wallace, also had a major insect-transmitted disease that he picked up in Indonesia when he was collecting there over the years. He had malaria, and it was, he writes that it was in a malarial fever that he came up with his independent idea that evolution was a real thing and that worked by natural selection. So the two founders of this science both probably had insect-transmitted diseases that affected their work. So without further delay, I'd like to introduce tonight's speaker. It's my pleasure to introduce Dr. Jason Rasgun, who's a colleague of mine and a friend of mine. I've known him for quite a long time. Dr. Rasgun got his PhD in entomology at the University of California, Davis, with his advisor, Dr. Thomas Scott. He moved on to a postdoc in entomology at North Carolina State University, where his advisor was Fred Gould. He took his first faculty position at Johns Hopkins University in the Malaria Research Institute there, and then moved on to his current position at Penn State University, where he is professor of disease epidemiology in the Department of Entomology, a member of the Huck Institutes of the Life Sciences, and the Center for Infectious Disease Dynamics at Penn State University. So please help me welcome with virtual clapping, Dr. Jason Rasgun.
Well, thank you very much for that introduction. Am I able to share screen? Let's try it. Okay, did that work? Can you see my slides, Dave? Yeah, okay. All right, great. So it is my pleasure to be speaking with you today. My only regret is that due to circumstances that I'm not able to be there in person. But hopefully at some point I will be able to. So today I want to talk about sort of the evolutionary processes that govern and affect invasive vector-borne diseases and vector-borne pathogens. Now I could sit here and talk for hours on this topic. I'm not going to do that, I'll spare you. So I'm really going to narrow down the focus of the talk today. And so I could be talking about malaria parasites, I could be talking about filarial worms, but I'm going to focus this talk mostly on, or exclusively on viruses, specifically arthropod-borne viruses or arboviruses. And this is defined, this is a term defined by Will Reeves decades ago, as a virus that can infect arthropods that inject infected vertebrate blood. It multiplies in the arthropod tissues and is transmitted by bite to susceptible vertebrates. So we're not really talking about pathogens or viruses that are just sort of mechanically transmitted by dirty mouth parts. There's an intimate biological association between the virus and the insect vector. There are many medically important arboviruses out there. There's probably about 400, maybe even more now that have been described. They're the greatest cause of viral associated encephalitis on the planet. There's maybe about give or take 100 of these that infect humans. There's maybe another 40 that are important for livestock. The rest of them are infecting wildlife primarily. Some of them are what are called insect specific, where they infect the insect, they don't really do anything else. There's a lot of different arthropod groups that transmit these arboviruses, but mosquitoes are by far the most important in terms of numbers. And we live in a connected world. So mosquitoes and vectors get moved around all the time. And so this gives opportunity for new invasions of spread of pathogens into novel environments where evolution can really happen. And I'm not going to talk about all the arboviruses because, again, I could be here for hours or days. I'm really just going to focus on West Nile. And this for a couple of reasons. Number one, this was actually the first system that I started working in as an independent faculty member. So it's kind of near and dear to my heart. Also, we're in Pennsylvania, at least I am, and at least those of you at Duquesne are. And if you're going to be infected with a mosquito-borne virus in Pennsylvania, it's almost certainly going to be West Nile virus. People were concerned when Zika and those things came through a few years ago, but your chances of being infected with Zika virus in Pennsylvania are virtually nil. Your chances of being infected with West Nile are considerably higher. So West Nile is called West Nile virus because it was originally isolated in the West Nile region of Africa, of what is now Uganda, in 1937. So we've known about it for quite a long time, almost 100 years. It was introduced into the United States in 1999, and in about 4 years, it spread all the way across the continent. And this was right in the middle of when I was in graduate school. So this was a very exciting time to be a medical entomologist. It was something, you know, you read about in textbooks that's happening right in your backyard. It's a very complicated cycle. There's over 40 mosquito species that have been implicated in transmission. At least if you bring them into the lab and give them virus, they can transmit, or you can isolate virus from them in the field. So it's not like dengue virus where it's basically 1 or 2 mosquitoes that transmit. There are many different mosquitoes that can transmit West Nile. And there are many different vertebrate hosts or reservoirs that can host the virus. So again, it's not like dengue where really humans are the only vertebrate hosts. There are many, many vertebrate hosts that are competent for West Nile. Birds, small mammals, even amphibians and lizards. Crocodiles can maintain West Nile virus. So it's a very, it can exist in very diverse vectors and very diverse vertebrate hosts. And it's the most important mosquito-borne virus in the USA. It's not the most important virus, perhaps, but it's the most important mosquito-borne virus. So with West Nile, again, most of the cases are asymptomatic. So about 95 to 99% of people that get infected with this won't ever know they had it. Or maybe they'll think, oh, I had the flu or something. So it can be, if you do get sick, it can be flu-like, but it can be more serious. So it can lead to rash. And in the really serious cases, where it'll infect the central nervous system and cause encephalitis, and then there can be rare fatalities associated with it. And these are more common in the elderly or the immunosuppressed, so people that are undergoing cancer therapy and things like that. So if they have a suppressed immune system, they're more likely to have serious disease. I just ran the numbers today. There have been about a little over 50,000 confirmed human infections and about 3,000 deaths from West Nile in the US since it was first introduced here. This is probably about a 10-fold underestimate because most of the cases of this are never diagnosed. These cases here are where they actually took blood and sent it to the CDC and it was a confirmed infection. So it's a vast underestimate, but it's the best data we have at this time. There's no vaccine or treatment available for public use. There are a few experimental vaccines out there, but nothing that you can go to the pharmacy and get. So really the only way to avoid it is to avoid being bitten by the mosquitoes that carry it. This is just showing the distribution of West Nile virus cases in the USA, data from the CDC per year from 1999 to 2021. Those 2021 numbers are provincial at the moment. They haven't been confirmed. But you can see it, the first year it was just trickling around for another couple of years and then it spikes. And this spike is probably less due to a massive increase in virus and more due to the fact that we were looking for it at that point. We weren't really looking for West Nile in 1999. And you can see we have some years that are higher than others, some years that are lower, but it's here every year. It's endemic, it's never going away, it's here forever. This is cumulative cases of not disease, but neuroinvasive disease. So this is serious disease where somebody presented with a infection of the central nervous system. Again, cumulative from 1999 to 2020. You can see there are definitely hotspots. And I'll come back to this, especially here in the Midwest Great Plains area. Really high levels of neuroinvasive disease. This is where you had really high levels of disinfection in general. And towards the end of my talk, I'll come back to some of the reasons why we think this might be. So just keep it in mind at the moment. And again, like most things, the disease tends to be seasonal. It starts peaking, around the summer, peaks in sort of late August, early September, and then dies down again. And this is coincident with the biology of the mosquitoes, when they tend to be active and biting, which it makes sense. You would expect this. Here's just a brief outline of my talk. I'm first going to talk about West Nile virus evolutionary adaptation to vectors and hosts in the novel North American environment. So again, West Nile is endemic to Africa and Europe, and it was really only introduced into North America, a little over maybe 23, 24 years ago. So this is all sort of a new open environment for the virus. Then I'll talk about some of the mosquito population characteristics that might have actually mediated this invasion of virus across the USA. And then finally, I'll talk about some of the factors influencing mosquito transmission of West Nile. And this first part of the talk is not our data. It's data from colleagues of mine, primarily at the Wadsworth Center, Laura Kramer's lab. These second and third parts of my talk are data that my lab has generated over the last 15 years or so. So West Nile is a flavivirus. It's got very typical flavivirus organization. If you're a virologist and you look at that, you would say, yeah, flavivirus. So I'm not going to go into it in detail. Just to say it's a positive sense RNA virus, which means that the RNA, the genome of the virus, can actually act as a messenger RNA in cells. If you transfect the RNA into a cell, you get virus that comes out. When we work with this in the lab, the RNA is considered infectious, just the same way that the actual virus particle would be. And And this is a very simplified life cycle. Remember, I said there's actually many vectors and many hosts, but I've just sort of simplified it down. In general, the virus circulates between mosquitoes and vertebrate hosts, birds, mammals, whatever it is. Occasionally, these mosquitoes will bite people or also horses, where they can develop severe disease. Both people and horses are dead-end hosts for the virus. So any evolutionary trajectories associated with transmission don't happen in us, even though we get sick, and don't happen in horses, even though they get sick. It's because we never raise a high enough level of virus in the blood to actually infect mosquitoes again. So virus that gets into you is, evolutionarily speaking, dead. It's never going to go anywhere again. It's not going to infect another mosquito. But it's important to us because it can cause disease. But the evolutionary trajectories are happening here in this mosquito bird cycle or vertebrate host cycle. Again, lots of mosquitoes that transmit the virus, but culex are probably the most important. So these are culex pipiens larvae. There's a culex pipiens adult laying an egg raft. This is also important because West Nile and some culex species can be transmitted from mother to offspring. So she'll lay eggs where the eggs are already infected with virus. And so when they hatch, they don't even have to bite somebody or bite a host to pick up the virus. They already have it. They can transmit it on the first bite. And that can be important in terms of the transmission dynamics. So this active transmission and cycling between mosquitoes and hosts actually has some very interesting dynamics in terms of the evolution of the virus population. The virus is not homogeneous. So a virus goes in, it replicates, it mutates to many different sort of viral variants. It's called a quasi-species. And it's a population of different viral genotypes. It's not unique to West Nile. This happens with pretty much every virus and even bacteria. West Nile virus is an RNA virus, so there's no proofreading in the replication, which means it has a really high mutation rate. So when you get a mutation, there's nothing to fix it. It just sort of stays that way. And these viruses have to be passaged between very different host types, invertebrates and vertebrates. So there are genetic bottlenecks at every step, which allows for different sort of drift events, which can fix different viral variants. And then simplistically, it kind of works like this. You have a host that's infected with a particular viral genotype. That virus mutates and as it replicates, and you get a population of viruses. Here, each color represents a different viral genotype. When the mosquito, that's your quasi-species, just this population of related genotypes. When the mosquito picks it up, it's not going to pick up all those genotypes. It's going to pick up just a random selection of them. There's your bottleneck. So the genetic diversity of the virus gets small again. When the virus replicates in the mosquito, it mutates. Again, you get another quasi-species. When it's transmitted to the host, only certain genotypes are transmitted again, another bottleneck. And mutation again. And so over time, you get these viral genotype switches from, let's say, the blue genotype to the green genotype. This can be driven by selection. Maybe certain genotypes replicate better than others. It can also just be due to what's called random drift, where just from selecting different genotypes every passage, you manage to shift the population over time. And that's what's happening here. We look here, this is a phylogenetic tree of different viral genotypes. What I've circled there is called New York 99. That was the first West Nile genotype to ever be sequenced. You can see it kind of fits in here. And these other ones, you have these at the bottom, the old world isolates. So the isolates that came from Europe. And then here in the middle, you have these isolates that popped up in the late 90s, early 2000s in the Eastern US. But starting from about 2002, you began to get this group of viruses here. And basically they eliminated all those early genotypes. So we had essentially, excuse me, a population replacement. So one genotype of virus essentially replaced all the other ones in the population. So after 2002, you never really saw these 99 type strains anymore. And if we lump all the 99 type strains together and all the 2002 type strains together, we can actually plot that. And again, this is not my data. This is data from Laura Kramer's lab at Wadsworth, but you can see in 99, all you saw was 99. In 2000, you saw the 99 strains. 2001, late 2001 is when those quote unquote 2002 type strains start showing up. And by 2004, they had completely replaced the original strains. It was a strain replacement event. And so now you find new genotypes now, but they're all related to this 2002 strain. So why did that happen? Was it random or was it something else? Were there some selective forces going on? It turns out the 2002 strains actually reach higher titers in the mosquitoes than the 99 type strains. They replicate better in the mosquitoes. And this is a log scale. So when you're going from 4 to 5, you're actually going 10-fold higher. So these 2002 type strains reach about 10 times the amount of virus in the mosquitoes than the 99 type strains do. And it turns out they actually infect the mosquitoes at a higher rate. So when you feed mosquitoes, this is Culex tarsalis, on virus, a greater percentage of mosquitoes over time get infected guts. This is looking at the virus, the proportion of mosquitoes that were infected where the virus actually gets out of the gut, which it has to do to be transmitted. So they have much higher rates of getting out of the midgut, and they have much higher rates of actually infecting the mosquito's salivary gland so it can be transmitted to another host. So these strains, they replaced the 99 strains. It wasn't random, it's because they're actually transmitted more efficiently by the mosquitoes. So they're selected what's called positive selection. And over time, they just outcompete the old original strains. So it's better adapted to the mosquito vectors than the original strains. And you expect this. You get a virus that's never seen this environment before. It gets in, it's maladapted. And over time, it has a very high mutation rate. It adapts to the local vectors so that it can be transmitted better. So it grows better in the mosquito. The 2002 strains grow better in the mosquitoes than the 99 strains. They're transmitted faster when orally infected. Actually, if you inject it into the mosquito and you bypass the midgut, it doesn't make any difference. So this speed of transmission is related to how the virus initially infects the midgut. So it interacts differently with the mosquito midgut. And faster transmission equals better transmission. I won't go into the math of this, but actually if there's an exponential relationship. If it's transmitted twice as fast, it's actually transmitted 4 times as efficiently. Or if it's transmitted 3 times as fast, it's transmitted 9 times as efficiently. So there's very strong selection for fast transmission, as fast as it can possibly be. So another interesting question is similar adaptation occurring in the vertebrate host. This is work from Aaron Brault's group, and they did this work in crows. Aaron knew for a long time, Aaron's a very good virologist, and he's done this not just with West Nile, but with lots of other types of viruses. They can get different isolates of West Nile going back all the way to the 1950s, and they can test them in animals, and they can say some of them are more virulent, they're more deadly to the host. And they can sequence the viruses and say, hey, they differ genetically. And it turns out when he looked at these sequences, there was one particular mutation that just kept popping up over and over. It was at the site amino acid 249 in the West Nile genome. And it was an amino acid threonine that mutated to a proline. And he found this in lots of different isolates, but they weren't related to each other. This mutation kept popping up independently. And it seemed to be related to West Nile outbreaks. It was variable geographically, it popped up all over the world. And he was wondering, is it involved in virulence or adaptation of the virus to the vertebrate host? You look here, these are just different isolates from Africa, Asia, Europe, and even Australia, going all the way back from the original Uganda strain in 1937, which actually had a histidine at that location, which we're not really going to talk about. But most of the strains generally have a threonine. So Tunisia, Italy, France, Kenya, China, Ethiopia. But then you have this proline mutation that keeps popping up, and they seem to be associated with outbreak strains. So again, even the New York 99 strain had that. Mexico, Israel, Russia, Australia. Actually, that had an alanine. But again, these were outbreaks that had this proline mutation instead of a threonine. So Aaron is a very good molecular virologist. So what he did was actually test this. He made infectious clones of the virus. What that means is you can take the whole viral genome and put it in a piece of DNA and basically make whatever changes you want to it, and then grow up that virus and test it. So I just want you to look at these solid lines here. And this is in birds, in crows. So he took these viruses and injected them into crows. So the blue line is a natural virus, a wild type virus that has a threonine at that site. And the red line is an isolate that has a proline. And you can see the proline one seems to replicate much better than the threonine. It's reaching what, 1, 2, 3, 4, 5, 6 logs, 7 logs higher virus, which means it's going 1 million times or 10 million times higher than that wild type strain. And then what he did is he took each of those strains and then he made one change. So he took that threonine virus and all he did was change that threonine to a proline. And then he can take, and so that takes this solid blue line and moves it over to the dotted red line. But at every other site in the genome, they're the same. It's just difference by 1 nucleotide. And you see immediately you get, 1,000 or 10,000 or 100,000-fold higher virus just due to that one mutant. And he also did the opposite. He took that proline strain and switched the proline out for a threonine and immediately attenuated it. So this difference in viral titer is solely due to this one mutation in the genome. And the other thing, when you look, this is actually the number of crows that survived. You get the exact same effect. So with the low titer infections, the crows generally do okay. They mostly survive. But the high titer infections, they die. And again, it doesn't really matter what the major genotype of the virus is, only whether there's a threonine or a proline at that site. So it shows you that threonine to proline mutation, it's an adaptive viral mutation. It's been selected for positively. The isolates with a proline there reach much higher viremia, and they kill crows a lot better than isolates with the threonine at that area. And you can engineer this in the lab, and these engineered viruses show the exact same profile as the natural viruses. This mutation has evolved independently multiple times, and when it evolves, the viruses generally do better and you get an outbreak. Higher viremia in the host also leads to much better infection of the mosquitoes. So again, it ties back to the vector, where these viruses also are much more easily picked up by mosquitoes because the probability that a mosquito becomes infected is in some sense dependent on the amount of virus that they feed on. They feed on more virus, they're more likely to become infected. And it's positive selection on a viral genotype. So these data show that West Nile virus adapted to this novel environment in North America. It was introduced in 1999. It was ubiquitous by 2003. The virus adapted to both the native mosquito vectors and the native vertebrate hosts. It's evolution for virus that's transmitted more efficiently by the mosquito and reaches higher levels in the vertebrate. And this, to me, as a student, was really interesting and cool because it was observation of pathogen evolution in real time. So, on the question of years, less than a decade, rather than sort of the hundreds or thousands or millions of years that you normally think of when you think of evolution. It was very, very, very quick. Okay. So I want to now go into this a little bit more. This is what sort of just showing how virus spread across the US. So this is data directly taken from the CDC again. And in 1999, you really just had it very localized in the East Coast, mostly Massachusetts, New York, and Maryland, and a few other places. 2002, it starts trickling up a little bit more. It's in New York, Pennsylvania, Virginia, North Carolina. 2001, it moved down to Florida. This is where we actually had our first major outbreak of West Nile, right there. I think that's Tallahassee. And it begins to move over a little bit more. 2002, it jumped. It made a major jump, basically from the Mississippi River all the way across the Great Plains. And then it kind of stopped, which was interesting. 2003, we had massive, major outbreaks in the Midwest. Remember I showed you that map showing neuroinvasive disease distribution. This is where all of that major neuroinvasive disease came from these massive outbreaks happening in the Midwest. And basically, it was almost just about hitting the West Coast. And by 2004, it was basically everywhere, pretty much every state. 2005, kind of the same way. And it's basically been this way ever since. It just sort of pops up everywhere. Something that was interesting to me when I was a brand new baby assistant professor was how did it make this jump? And so there have been a few modeling papers out there where people said, oh, birds moved it. But actually, when you look at their data, the data were kind of crappy. It didn't actually make any sense. The birds tend to fly, the flyways are north-south, not east-west. So I could believe potentially the birds would move it from New York to Florida, but not from Florida to California. So we began to look at this a little bit more. I'm an entomologist by training, I'm biased, but we began to look at mosquitoes, and we ended up focusing on this particular critter, my favorite mosquito, Culex tarsalis. Very beautiful mosquito, just gorgeous. Nice to work with in the lab. It's just a wonderful thing. It's a very important general vector of viruses in North America. It transmits a lot of different things, both to humans and also to wildlife. Transmits to humans St. Louis encephalitis, and Western equine encephalitis. Turns out it's one of the most efficient vectors of West Nile virus ever seen. It also transmits a lot of other things that I won't go into. So we began to look at the populations of this virus. And so again, this is work we did a long time ago, but it's still relevant. Where we actually had, we were collecting adult mosquitoes across the US. Basically, this is the entire pretty much distribution of this mosquito across the US. It doesn't really occur very much east of the Mississippi River. It's a Western US mosquito. And so these collections span the range of tarsalis in the USA. We had 20 populations in 6 states. We used microsatellite loci. Don't worry about the colors here. It doesn't really mean that much for this purpose. And just briefly, what microsatellites are these regions in the genome where you have these repeated regions like AT. You design primers to amplify those regions across, and depending on how many repeats you get, the size of the fragment you get is different. And then you can run this out on a gel, or here we're running it out on a sequencer, and you can see different peaks for each of those alleles. And so you get a bunch of marker profiles. We had 12 of these different ones. So we could actually get quite a lot of markers for mosquitoes. And we can use these markers to compare the similarities of different mosquitoes. How similar are they genetically? And I can go into this more if you're interested later. It's just a way to look at the genetic relationship among individuals. And these work pretty much in any organism. And without going into a lot of the details, we actually found 3 different groups of populations in the mosquitoes. And so one we found was sort of all the Midwest and Great Plains clustered together. Then we had sort of everything west of the Sierra Nevada mountain range. And then we had this weird little cluster in sort of Southern California, Arizona, the Sonoran Desert. Looking at this on a map makes a little bit more sense. So here in yellow, we have the Great Plains populations. These all come together as sort of one big meta population where mosquitoes are kind of moving freely between them. We have this Western intermontane and coastal region in red, in purple here too. And then we have the Sonoran Desert right here at the bottom. And this is just this exact same population, just showing the clades of genetic relationship, where we got this map from. And when we got this actually looked very familiar to me. And it turns out that when you overlay this is the exact same data I showed before. The 3 different clusters. Tarsalis doesn't really occur over here on the East Coast, but you've got your Great Plains region, your Western Montane region, and your Sonoran Desert region. In 1999, this is the mosquitoes here, or West Nile here. 2002, this is probably being driven by other mosquito species, probably Culex pipiens. 2001 is when the virus actually hit on the easternmost border of the Culex tarsalis species range. And in 1 year, completely jumped across, but it stopped here, which is interesting. In 2003, it began to trickle over and it moved over into the Sonoran Desert region. So it went this way, kind of over the mountains and then under through the Sonoran Desert. And then by 2004, it was everywhere. And then again, nothing's changed at that point. This is basically still how the situation looks today. So tarsalis seems to exist as 3 metapopulation clusters, where you have basically free movement of mosquitoes between populations within a cluster, but restricted movement, although it's leaky, between the clusters. So it basically stops at the cluster border and then leaks over rather than being freely moving. And the very interesting thing, the virus movement mirrors the mosquito population structure. When you actually look at correlation statistics between the mosquito population structure and the patterns of virus movement, it's about 96%. So it's an order of magnitude better fit to the data than the bird models that were out there. So this is what we think is actually happening. We actually think that the Tarsalis moved the virus across the US. So this was actually the first evidence for mosquito-mediated dispersal of really any mosquito-borne pathogen in any geographic area. There have been some work now done with Aedes albopictus and other pathogens that have kind of shown the same thing. So up until this point, mosquitoes were kind of ignored as potential vehicles for pathogen movement across large spatial distances. And now it seems to be, this is something that does happen. So then you say, well, how far does the mosquito really move? It depends on the mosquito. Aedes aegypti mosquitoes, which transmit dengue virus, if they move 100 meters in their lifetime, that's a lot. But Culex tarsalis move, especially host-seeking females. They travel for consecutive nights. So they'll move on one night, and then more on the next night, then more on the next night. And even in one night, if the winds are right, they can move up to 4 kilometers. So over, the mosquito's lifespan of, let's say, 20, 30 days, they can move, hundreds of kilometers. So, it's the same amount of movement as the resident bird populations. And they also disperse randomly to hunt and feed. So it's not like they're following corridors of movement. It's sort of, can be modeled as a random diffusion model of, particles moving over time, because that's kind of how they do things. And then this can be mediated even more by airflow wind, and they can get picked up in cars and trucks and moved. So they can move quite a distance. This is probably how West Nile got to the US in the first place. It was probably on a mosquito that got onto an airplane or something like that. Okay, so then we started to get really interested in Culex tarsalis, and I'm going to move into the third part of my talk now. So this is, remember, this mosquito has never, up until maybe 2000, 2001, had never seen West Nile virus. It did transmit a related virus, St. Louis encephalitis virus, but it never saw West Nile. So the question was, what is governing or making this mosquito such a good vector if it's never seen it before? What are the factors that are being selected on or driven to allow transmission? Maybe it's just the genetics of the mosquito. The jury is still out of this. Our initial studies actually showed no evidence for this, but with the sort of sequencing technology has really improved dramatically in the last 15 years. And so has experimental design to do inheritance and things like that. So we're actually looking at this right now. So I'll say we don't know yet. There is probably a host genetic component. Is it environmental conditions? Sometimes you can rear mosquitoes at different temperatures or with different amounts of nutrition, and you can change the way they transmit pathogens. We tested this with Culex tarsalis, and it didn't really matter. It didn't matter what temperature we reared them at. It didn't matter, how starved or well fed they were. They all transmitted the same. So environmental conditions didn't seem to matter. Turns out the biggest thing that we saw that had an effect was the microbiome, or the bacteria essentially in the mosquito's gut. Had a huge effect. So what do I mean by microbiome? You have a mosquito. Mosquitoes, like all organisms, don't exist in isolation. They're a conglomeration of cooperating organisms. So you have, if without your gut microbiome, of the bacteria in your gut, you would be having major problems. Mosquitoes are the same way. So you have different bacteria in the gut, and different mosquitoes, when you collect them and you test them for what bacteria they have, there's variation in the bacteria. And so you can have certain mosquitoes where certain bacteria get in there, they can kick out other bacteria, you can get competition. This can change the way bacteria replicate, and it can change the distribution of bacteria. And if the bacteria in the mosquito actually determine whether or not they become infected with virus, patterns of microbiome variation in the field can actually have a strong selective effect on whether the virus is transmitted or not. We found this out by accident. So you look at the number of days here. This was an experiment, took a little over 6 years. What we were originally trying to do was actually determine the genetic components of West Nile vector competence. So we took mosquitoes and we actually, in the lab, selected them to be able to transmit West Nile, which are resistant lines, or not transmit them. The green line here, susceptible. This is a big pain because West Nile is a biosafety level 3 pathogen. All this work had to be done under high containment. You see how long we had to do this for. And we thought we had things going pretty well. We finally had, a pretty reasonably resistant line of mosquitoes and a pretty good, consistently susceptible line of mosquitoes. I'm not going to show it, but we actually did this large genetic genetic mapping experiment. Turns out we got no significant genetic signal of West Nile vector competence. And that was weird because you have such a strong difference here. And what clued us in that we were looking at the wrong thing was we had this case here where our resistant line, in like maybe 1 or 2 generations in the lab, all of a sudden became completely susceptible. So this red line here and this blue line are the same mosquito line. It just, after many, this is 3-4 years of resistance, it became susceptible. This was coincident with switching the mosquitoes to a new insectary. So we realized, maybe what we're seeing, this phenotype that we're seeing is not due to the mosquito genetics. Maybe it's actually due to the bacteria, which they pick up in the insectary environment. We were able to select them again for resistance much faster than before. When we stop selecting, they start reverting again to susceptibility. And again, this is much faster than you would expect if it was the host genetics responsible. Luckily, we had saved mosquitoes from all these lines at multiple time points. And this was right when sort of the sequencing technology hit the point where it was relatively straightforward to start assaying in a high throughput manner what the bacteria were in your samples. So I'm generalizing and summarizing a massive amount of data here into one graph, because we did this with multiple different independently selected mosquito lines and different experimental conditions. I'm just showing sort of what fell out of all of that. So we have Culex tarsalis, where we sequenced a region of the bacterial genome called V4. Don't worry about that. And basically, these are the bacteria families that are enriched in the resistant lines. These are the ones that are enriched in the susceptible lines. And what we find, the susceptible mosquitoes were highly enriched in a bacteria called Erythropetingia, which I'm not going to talk about, but it's kind of interesting in of its own right. And the resistant mosquitoes were highly enriched with a bacteria in the family Acetobacteriaceae. When we looked into that a little bit more, it was a bacterium called Asaya. And this is a bacteria that Dr. Lampe's worked quite a lot with. So we started to look at this a little bit. It turns out if you take Asaya and you introduce it into the mosquitoes in their sugar or blood meal, and then you give them a West Nile infected blood meal, their ability to become infected with West Nile is reduced by about 50%. So it seems that our model here is that at least we know in the laboratory, Asaya seems to be governing whether the mosquitoes become infected with West Nile or not. This is actually also sort of feeds into other research we're doing in the lab independently. We actually isolated this bacteria, Asaya, and its relationship with West Nile independently in 2 completely unrelated experiments that we were doing for completely different reasons. So we're pretty confident of this result. So it turns out that West Nile susceptibility, it's not just due to the genetics of the mosquito or the environment. It actually changes with the composition of the mosquito gut microbiome. And this is independent of the mosquito genotype. Again, I'm not showing this, but we actually had multiple lines of mosquitoes from different origins with different genotypes where we saw the same effect. When you select for resistance to West Nile, the microbiome becomes enriched for Asaya. So you're actually selecting on the gut microbes rather than the actual genotype of the mosquito. And it's Asaya we found in these lab studies to be associated with increased West Nile resistance. And Asaya supplementation to the mosquitoes that makes them resistant to virus. This is a laboratory study. I have no idea whether this is happening in the field, whether variation in microbiome is conditioning variation in vector competence and West Nile transmission under field conditions. We are currently looking at this. I have a funded NIH grant to do that. So to conclude, I'm just going to wrap up here so we'll have time for questions. Pathogens get moved around the world all the time. I've been talking about West Nile, but I could have had this talk and talked about Zika virus. I could have talked about Chikungunya virus. I could have talked about Venezuelan equine encephalitis virus. The same sort of processes are driving the evolution of these pathogens. No matter what the pathogen is, it's the same kind of dynamics. And pathogens in a new environment adapt quickly. So on the order of a decade or less. So this is blindingly fast in evolutionary terms. It's very surprising to me. And just because the new native vectors and hosts, just because it's a new pathogen, the native vectors and hosts may still be pretty competent. They might even be more so than the hosts and the vectors in the originating area, because there's no resistance to these pathogens yet. And then they can evolve to become even better over time. And then the spread can be rapid. It can be facilitated by properties of the new environment. And evolution can happen at multiple scales, not just at the viral level, not just at the mosquito level, but it's this concept we call the hollow genome, which is the genome of the organism and all of its associated microbes. So evolution can happen at all these scales all at the same time. It makes this kind of questions very complicated to study, but also very, very interesting. With that, this is not even everybody that's helped with all these studies. It's a lot of people. I really do want to highlight Brittany Dodson here up in the upper left-hand corner, who did the bulk of the West Nile work, and also Marco Brusselin, who did a lot of the Asaya work. This was the bulk of Brittany's PhD thesis a few years ago. She's also a professional science writer. If you guys need any help, she's the person to go to. And then finally, we've been very lucky to be funded by many different people and groups and organizations over the years to do this and other things. Primarily, most of the West Nile work has been funded by the National Institute of Allergy and Infectious Diseases at NIH. And with that, I think I'm just about done. I can take questions. Thank you.
Well, thank you very much. You can't hear the rest of the audience, so on behalf of the over 200 attendees, I'm clapping for you. Yeah, I've read the questions here. I'm going to kind of combine a few of them at the same time. First of all, you mentioned a little bit about how the virus got to the US. You suspected it might be a mosquito on a plane. How sure are we of that, and what evidence, or how do you go about determining that?
We're not sure of that at all. That's my gut telling me that, because it's easier for a mosquito to get here rather than an infected bird or something. It could have been a bird that flew all the way over the Atlantic, but then you have to wonder why didn't it happen before. I guess you could say the same thing with the mosquito. But my personal gut is that it was a mosquito, but I should say I don't have evidence for that. That's a supposition. And I might be biased because I'm an entomologist.
Could it have been a human?
Probably not, because humans are dead end hosts for West Nile. So if a person is infected with West Nile and a mosquito feeds on that person, the mosquito won't become infected. Humans just don't have a high enough viremia in the circulating blood.
And as part of the follow-up to that, person asked, why did it take so long? If West Nile has been in Africa since 100 years or more, why did it take 1999 to finally get to the US?
So that's an excellent question too. I don't know. There's a possibility that it has, but some of these things, they're kind of like sparks. So they go and they spark, and sometimes it starts a wildfire, sometimes it just burns out. It's quite possible that it had been introduced here before and either wasn't recognized and petered out before it spread, or just for whatever reason, we got lucky, so to speak. Those are difficult questions to answer.
Well, that's why we're asking them.
Yeah.
So question about the 2002 strain, a couple of questions. Did it replace previous strains just in the US or around the world?
That is an excellent question. I do not know the answer to that.
And the other part of the question related to that, if this particular mutation of 3-neoproline increase the death of birds, is that counterintuitive while we often think about hosts or viruses evolving to become less virulent as to not kill off their hosts?
So that's actually a misunderstanding. Viruses are not necessarily selected to become less virulent. They're selected to be transmitted more efficiently. So if a virus is transmitted more efficiently but kills the host, you will select for that until all the hosts are dead. So, you know, in those cases, it's killing the crow, but in the process of killing the crow, it's reaching 1 million times higher viremia in the blood. Mosquitoes are much, much more likely to become infected as they feed on those crows. And the crows are also sick, so they're not moving around and pushing the mosquitoes away. So definitely you can get selected for that. Over time, actually, we don't see these days the kind of bird deaths that we saw when West Nile first was introduced here. And that's because the native, you know, over time, the host will eventually evolve to mitigate the fitness costs of the virus infection. But those initial selective forces, the virus is being selected to be transmitted, not to become attenuated in the host.
Thank you. A couple questions here related to mutation rates. First one coming from someone who says, I'm not a virologist. How can West Nile virus mutate so rapidly? Is it common for other viruses to have such high mutation rates?
Yes, it's extremely common, especially for RNA viruses.
Okay, and then this question related coming from an evolutionary biologist, how large is the West Nile virus genome? And given its mutation rate, how long does it take to sample every possible mutation?
Okay, so the genome is about 11 kb, about 11,000 nucleotides. I don't know how long it would take to sample everyone. Nathan Gruba at Yale, who has done a lot of work of sampling and sequencing viral genotypes, could probably answer that a lot better than I could. I could probably sit down and calculate something, but I don't know off the top of my head how long it would take.
Okay, and quick question, what keeps Culex tarsalis out of the eastern US?
So when I say it doesn't occur here, that's not entirely true. It does occur every blue moon. You'll find one in a collection, but we don't have sustained stable populations. I don't know why that is. Honestly, it probably could be exclusion maybe from other mosquito vectors, but I don't really know. Like, it's not really going to establish here in Pennsylvania. It gets too cold in the winter. I don't see any reason why it couldn't establish in Florida, but it doesn't seem to. So I don't really know. I don't know why. But occasionally, like if you look at the collection records, it'll pop up occasionally on the East Coast. It's not like it is in California, where I can stick out in Sacramento a mosquito trap and get 10,000 tarsalis in a single trap night.
Okay. Someone says they recently read in local Pittsburgh media that I believe it's a new highly infectious virus that is transmitted by deer ticks. Do you know what this is? Can you tell us a little bit?
So it's not really new. It's Powassan virus, and there's a very, very, very closely related virus called deer tick virus. They're both in the tick-borne encephalitis group of flaviviruses. It's not new. It's been around forever. It's been historically rare. So generally, in a given year, you'll get a handful of cases, like 2, 3, 4, 5. We've been seeing lately in the last decade or so an increase in incidence. So it's still very rare compared to other pathogens, but it's still greatly increasing relative to how it used to be. And that's a nasty one. So again, it's rare, but if you do get it, it's got something, I forget exactly what the numbers are, but it's something like a 40% death rate. So all the tick-borne encephalitis viruses, most of them are in Europe. This is the one that's here in the North America. They're all quite nasty. But so it's not new. It just seems to be expanding in incidence now.
Okay, and now I have a series of questions coming from what appears to be people who know a lot more about this stuff than I do, so I'll probably mispronounce words. There has been evidence that Asai excludes Wolbachia co-infection from Anopheles mosquitoes. Is there any evidence that this occurs with Culex mosquitoes? And how does that affect the ability for West Nile virus to infect Culex?
Yeah. So that's a great question. So my lab is the one that actually found that effect in Anopheles. So Culex tarsalis is not naturally infected with Wolbachia. They don't have it. We actually showed when you infect them in the lab, basically it does, Asai levels go way down. So we had published a paper. So normally Wolbachia is a bacterium. When you transfer it into mosquitoes, it makes them much more resistant to virus infection. And these mosquitoes, Aedes aegypti mosquitoes, have been released in something like 12 or 15 countries already. But we found a long time ago now that when we artificially infected Culex tarsalis with West Nile, they actually became more likely to become infected with I'm sorry, when we infected them with Wolbachia, they became more likely to become infected with West Nile. And it turns out that's because when you infect them with Wolbachia, it suppresses Asaya, which increases their susceptibility to West Nile. We're actually getting ready to publish that now. So it's exactly the same effect. And that's actually the other unrelated study where we got to the same relationship of Asaya and West Nile, where we found actually in these two different experiments, they both came down to the exact same bacterial isolate. So that's why we're pretty confident of the result. Again, it's a lab study. I don't know what's going to happen in the field, but that's definitely what we see in the laboratory.
Okay, and kind of related to that, just generally, what do we know about the mechanism for how the presence of Asaya results in increased resistance?
So I don't know that, but what I do know is that whatever's happening, it's happening at the initial event of virus infection of the midgut cells. So if the mosquitoes still get infected, so because it doesn't block 100%, then the virus disseminates to the tissues and it transmits by saliva at the exact same rate as the controls. So whatever it's doing, it's blocking or stopping that initial infection step of the virus to the midgut epithelium. And so my guess is, again, I don't know this. My hypothesis is that it's maybe blocking the receptor that the virus is binding to or something like that. It's hard to test that at the moment because we don't know what that receptor is. But that would be my hypothesis. Maybe it's forming a biofilm and it's blocking virus from getting to the midgut. I don't really know. But whatever's happening, it's at that initial infection stage. So it's not like increasing the immunity of the mosquito or to drive down virus level or suppressing the immunity to drive up virus or something like that. It's something about the way the virus is infecting the mosquito midgut.
Okay, a couple of related questions coming in from a lot of our favorite local Pennsylvania evolutionary biologists. Just want to put that out there. I'm glad we're using this event to build a greater community. I see some faculty here from Penn State Beaver and from Lehigh and from Mount Aloysius. But the question here is, any fitness costs or benefits to the mosquitoes infected with West Nile virus, or is it only the vertebrate hosts that have?
So with some viruses, you can detect fitness costs or benefits in the mosquito. To be honest, we have not tested that. So I don't really know.
Okay. Is there a potential for West Nile virus to become more deadly to humans in the future?
There's always potential. It's probably unlikely because, again, with humans, the virus is a dead-end host. So there's not a whole lot of evolution going on in terms of the viral interaction with people. People are not involved in the viruses evolved to be transmitted, and people are not involved in the transmission cycle. That's not to say that something could happen like in the bird cycle that could have spillover effects into people. But I think at this point in the site, it's been here for quite a long time, and I don't think that's likely. But again, anything's possible, but it's probably not very likely.
Okay, so you focused a lot on West Nile virus. Is that typical of other mosquito-borne diseases with respect to evolution of virus, potentially the involvement of the microbiome and the forces and processes? Generalizing.
Yeah. So a lot of these and again, in terms of like looking at particular viral genotypes that are selected for, again, Aaron Brault has done a lot of that work. He's done very nice work with dengue. He's done very nice work with Venezuelan equine encephalitis virus, where again, they can find isolates that are associated with virulence and outbreaks and ones that aren't, and they can get these down to single mutations and switch the mutations and show that, yeah, that's due to that. So he's done this actually for a lot of different unrelated viruses. And there's more and more studies coming out now showing that microbiome can affect many different mosquito-borne pathogens, not just viruses, some malaria parasites and things like that. So yeah, I think the West Nile, as I'm talking about it here, I'm really presenting it more as an example of a related group of systems that all kind of have the same general behavior. So I don't think West Nile is an outlier here. I think it's sort of kind of a poster child for these dynamics that are happening in a lot of systems.
Can you predict the future of West Nile virus and what we could expect from it?
I think it's going to be here as an endemic, quote unquote, virus forever, is what I think. And then, basically, if you actually look at I can actually have those data. Here. So in general, it seems that everybody can see that.
Yes.
It seems that the viral cases are going down over time. And I think a lot of that is due to, immunity. So again, in the bird populations, when the bird becomes infected, it's generally immune. And so you get, a greater proportion of immune amplifying hosts. And so the transmission is not as efficient. These initial spikes are partially due to just people starting to look for virus, and also due to the virus sort of, having a field day in a completely naive environment. And that's not really the case anymore. So really what you're going to have is a virus that kind of trickles along at some low level, and then every few years, maybe every 10 years, 20 years, who knows, you're going to have spikes as you get basically enough of a population of hosts that are not exposed anymore. And then it's going to trickle down again. There's a lot of sort of endemic viruses that act this way. Eastern equine encephalitis, St. Louis encephalitis, you kind of get that, where it trickles along and then you get a spike, and then it trickles along and you get a spike. I think that's what's going to happen. That's already what seems to be what's happening with West Nile.
So speaking of the future, and I don't know anything about hibernation or overwintering, as our climate gets warmer, and right now it's 60 degrees in Pittsburgh in February, will that increase mosquitoes and mosquito-borne diseases in the US?
It's hard to say if it's going to increase. It's going to change the distribution, because in areas that were too cold to maintain transmission might get warm enough to do it. But areas that were already warm enough to do it, they might actually get too hot. So mosquitoes and these viruses exist in a thermal optimum. So when it's too cold, you don't really get transmission. When it's too hot, you kill the mosquitoes. And so what we might actually see, rather than sort of a general increase, is a change in the distribution, where you start seeing virus in places where you didn't have it before, and you stop seeing it in places where you did have it.
Getting back to this 3 interpearline mutation, if it affects the titer in mosquitoes, does that make West Nile virus have more of an impact on humans or whatever the next host is?
So it affects the titer in birds, is where it's been tested. So it doesn't affect the titer in mosquitoes, other than mosquitoes, when they're feeding on those birds, are feeding on a lot more virus, which just naturally gives them a higher titer. I don't think anybody's ever tested what those do in people, because to do that, you'd have to actually infect people with West Nile, which you won't get permission to do. So I don't know that anybody's done that, even like monkey experiments, because people have mostly been focusing on birds, because birds, especially crows, corvids, were known to be amplifying hosts. They normally would get, very, very high titers of virus anyways.
Well, here's a general question. Do we need mosquitoes? Can we just bring back DDT in a controlled spray?
So again, DDT works for malaria control because of the way it was applied, especially for Anopheles mosquitoes. They would tend to feed on people and then rest on the walls of the houses. So you spray DDT on the wall of the houses, and that kills the mosquitoes. This is Culex mosquitoes, for instance, spraying DDT on the walls of your house is not going to affect West Nile, because that's not the way the transmission cycle works. You're being bitten outside. And you can't just spray DDT or other pesticides willy-nilly because of all the off-target effects. Also, I always hate when people say we should get rid of mosquitoes. There's something like 3,000 or 4,000 species of mosquitoes. There's maybe only 50 that really cause major problems. So most of the mosquitoes don't really do anything to people. And I love them, and we should keep them, personally speaking. So, if you have a disease like dengue, where it's essentially, for the most part, a single mosquito species that tends to be an introduced species anyways, and humans are the only hosts, you could eradicate those potentially. And there's a number of strategies people are trying to do that. For something like West Nile, where you have dozens of vertebrate hosts and dozens of potential mosquito vectors, I don't think you're ever going to get rid of it. There's no way to get rid of all the species of mosquitoes that transmit it. So I think we're stuck with it. Put on repellent if you go outside.
Okay. At least mosquitoes have one defender. in the audience today.
They also pay my mortgage, so I really like them.
Has any work been done on the impact of the microbiome on the vertebrate host susceptibility to West Nile virus?
I don't know about West Nile, but this is an active area of research. People have looked at the role of the human or mouse or whatever microbiome on lots of different pathogens, and it can have huge effects. So I don't think anyone's looked at West Nile particularly. I would not be surprised at all to find out that it had a major effect. But I can't think of any studies off the top of my head where somebody's looked at that. But it's also not my area, so I might be wrong on that.
This is a pretty specialist question, so you may have to explain the question to the general audience here. Is Culex torsalis an autogenous? Am I saying that right? And does this have any effect on its vector capacity?
So it's both. So it's autogenous, so autogenous means it can lay eggs without taking the blood. So female mosquitoes, they don't take blood because they want to hurt you. They take blood because they want to lay eggs. They need that protein. And so autogenous mosquitoes can actually lay eggs without taking a blood meal. Now tarsalis can be facultatively autogenous. So some mosquitoes, if you just take a bunch of tarsalis in the population, you bring them into the lab and you give them a place to lay eggs, some proportion of them will just lay a first batch of eggs without taking a blood meal. They need blood to take the second meal. We actually started working on the physiology of tarsalis. We were actually trying to find the mechanism of autogeny. It was quite easy actually to select for a line of tarsalis that was 98% autogenous. We never had to blood feed it. We ended up finding some very interesting things about how yolk protein was regulated. We never found the mechanism of autogeny. But a lot of Culex mosquitoes can be autogenous. If they need blood, that's called anautogenous. So they can be both. And definitely for the second batch of eggs, they're anautogenous. So if they don't take a blood meal for the first one, it decreases the biting rate overall, and that can decrease transmission. But tarsalis can also be infected vertically from their mother, which means they can still transmit on the first biting, so that can increase it. So where that wash lies, I don't really know.
All right, and we're switching to a different species here. Can you tell us a little bit about the CRISPR-Cas9, the use of CRISPR-Cas9 to reduce tick-borne diseases?
Yeah, so we just had a paper published. So at the moment, nobody's using CRISPR-Cas9 to reduce tick-borne diseases. That's because up until 2 days ago, there were no published accounts of doing genetic modification or CRISPR in ticks. So we and my collaborator, Monica Guglielmus and Andrew Noose at University of Nevada, Reno, and Rob Harrell at University of Maryland just published the first CRISPR tick paper. So this is now going to be opening up a very active area of research of using the power of genetic modification to potentially control tick-borne diseases. The problem with ticks is not so much the genetic modification, it's just they're, excuse my language, a pain in the ass to work with. The life cycle is really long. Like if you really push them, maybe you can get them to go through a complete life cycle in 4 to 6 months if you really know what you're doing. They're very difficult to rear. They need a blood meal every single stage. The larvae are smaller than a pinhead. So they're just technically very difficult to work with. Once we and our collaborators figured out the protocols to do the genetic modification, though, that was actually pretty efficient. It's actually more efficient than we see in mosquitoes. So ask me that question again in 5 years, and I'll probably have a better answer. But it's a very active area of research now.
Thank you. Here's an interesting question. Could you use mosquito probiotics to combat arboviruses, and how might that actually work in practice?
So that's a very interesting question. One of the things that we're looking at with our field studies would it be possible to sort of introduce bacteria into mosquito habitat to actually change their ability to transmit virus? And that's one of the goals of the study that we're doing right now. The first thing we're trying to do is actually just characterize across the species range of this mosquito, what does the microbiome look like? And also, we're not just looking at bacteria, we're also looking at fungal and viral microbiome, because there's a whole other suite of microbes in there. But yeah, it's possible. And there has been some work in that, particularly with using genetically modified bacteria, although it doesn't have to be genetically modified. And people have worked on ways to introduce that into the wild, especially using something called an attractive sugar bait. Because mosquitoes, even though they feed on blood to lay eggs, they still need sugar for the sort of day-to-day nutrition. So you can put these bacteria into basically little sugar stations, and mosquitoes will come and feed on that, and they can pick up the bacteria that can have an effect on pathogen transmission. The problem with that is you get other things, ants and butterflies and stuff that also like those sugar baits. And so mitigating non-target effects is a bit of an issue, but it's definitely an active area of research with a lot of different groups at the moment.
All right, attractive sugar bait sounds like my new band name. All right, so you said culex can feed on both birds and mammals, and is there a difference in how they metabolize nucleated versus non-nucleated blood? And could you maybe explain that to the audience, what we mean by that distinction?
Yeah, so mammals generally, except for very few, I think camels have nuclei in their red blood cells, but most mammals don't. Birds do have nucleated red blood cells. So there's definitely been a lot of work done, especially you go back to like the 1930s, 40s, and 50s, where people have looked at how mosquitoes do on different types of blood. And some of that work has been sort of nailed down to particular amino acid compositions of different blood. I don't think it's due to the presence of nuclei in the blood cells or not, but definitely, certain amino acids are at different compositions in the blood of different types of vertebrate hosts. And mosquitoes definitely can do better on some types of blood than others. Okay. And it's different for every mosquito, really.
I think I'm going to make this our last question. We're kind of hitting time, and it's a lighthearted question. Have you listened to This Podcast Will Kill You?
I have not.
You're not familiar?
Sorry.
All right. In that case, I want to thank you again on behalf of all the audience and for spending your time with us and teaching us a lot, including the importance of evolutionary biology in our modern world. So thank you. And thank you, everyone.
Thank you. Thanks for having me.